Great. Thank you so much for the opportunity to share more about the Aspect story, and special appreciation to the Alliance of Regenerative Medicine for giving us the opportunity to present. So I'm Tamer Mohammed, founder and CEO of Aspect Biosystems. Our company is based in Vancouver, Canada, and we're focused on creating what we call bioprinted tissue therapeutics. So these are living cellular therapies, and I'll tell you more about what those are. So what is a bioprinted tissue therapeutic? So these are implantable living cellular therapies that we develop allogeneically and implant into the body to replace, repair, supplement biological functions inside of the body. The way that we produce these tissues is by combining our computational tissue design with our microfluidic bioprinting technology, fit-for-purpose biomaterials, and at the heart of these cellular therapies, our allogeneic stem cell-based therapeutic cells. So by virtue of an overview to the company, Aspect was spun out of the University of British Columbia in Vancouver, Canada. We left campus in 2017, so we've been operating commercially for about 5-6 years right now. To date, we brought in over $150 million in capital. Our R&D is conducted in Vancouver. We have a team of over 90 scientists, engineers, and therapeutic developers. We have a broadly applicable platform technology, and so a big part of our focus has been to identify those initial programs that we're moving forward, and we have a big focus around metabolic and endocrine indications. And we're advancing our own pipeline, but we also recognize the opportunity to partner, and so we recently announced a partnership that we're very proud of with Novo Nordisk, and I'll be highlighting that later on in the presentation. So first, as an overview to our platform technology, so really what we've built at Aspect is what we call a full-stack tissue therapeutic platform that allows us to start the process with designing these tissues in a computational setting, where essentially we're making decisions around the overall tissue structure, the composition, the cellular components of our tissue. And then we move into the real world where we're taking the stem cells that are, of course, at the heart of our tissues and different fit-for-purpose biomaterials that have different functions, for example, providing a level of immune protection. So these tissues could be delivered allogeneically into the body without the need for immune suppression. Other materials that could encourage vascularization in the body. And of course, the materials that the cells are going to live in. Our problem is not just a delivery problem, but we want the cells to live in these tissues inside of the body. And so providing an appropriate home for these cells to live is a big part of our focus. So we bring in all of these different inputs, both the living inputs and the biomaterial inputs, into a microfluidic bioprinting technology that's proprietary to Aspect. And through computer control, we can mix and combine these different materials and then in real time essentially create our combination cellular drug product in a single step, and then ultimately package, cryopreserve, and ultimately deliver this into the body to provide a functional cure. And so at Aspect, we're really combining all of the necessary elements to vertically integrate this and deliver a complete solution. And so just mapping that out into sort of a real life type of a workflow, you could see how we're able to design these tissues in the computational step. And then you could see a close-up of the cells, in this case, a close-up of stem cell islet-like clusters. Then we move into the manufacturing step where we're creating these structures that are packed full of cells and the other types of materials that we incorporate into our tissues, and then we implant those tissues into different sites around the body that in many cases are remote to the organ functions that we're looking to replace or supplement. And so really what we've created is a multi-parameter optimization system to really maximize tissue function and replace malfunction due to disease. And so what you could see on the left-hand side here is a visualization of a representative bioprinted tissue. We could control the overall shape, the size, the density. But what's even more powerful is at the microscale, where a lot of the biological magic really happens, is we have a lot of control. So here's just an example where The cells, for example, are loaded in the core, and in this case, we're demonstrating islets in the core in a material that is designed for those islets to perform in what we believe to be an optimal way. And so you could see islets that are stained for essentially insulin, and you could see that these islets are functional and happy within that material. And then we have a further material that surrounds that core that provides a level of immune protection. And so this is a semi-permeable membrane that has a totally different function. And that function is to let things in that we want to come in and communicate with the cells, and let things that are produced by the cells come out of our tissue. So for example, glucose and insulin in the case of diabetes, but of course block any cells from coming in and facilitating a cell-mediated attack. And so here you could see a cross-section of that tissue, and looking at one of those individual fibers and looking at CD45 staining, you don't see any cell-mediated attack. And then we incorporate another material that is essentially designed to encourage vascularization and dampen the host, or optimize the host tissue response and dampen the foreign body response. And so really this is an example of how we're able to bring in multiple different materials in different architectures to solve a multivariable problem using this multi-parameter optimization system. The important thing to note here is this entire product is manufactured in a single step. So we're not creating cells and then a device and filling the device with cells. We're not creating a macro device that is subject to issues of cell viability, and we're not having to rely on a micro sphere that we would have difficulty retrieving. We're essentially able to leverage the best of the micro worlds with the best of the macro world in a single cellular combination product that's manufactured in the matter of minutes. So a human-sized tissue could be manufactured in as quickly as 10 minutes. And so this is a very streamlined approach and takes the better of many of the different approaches in terms of encapsulation and delivering allogeneic cell therapy that we've seen in the past. And so creating such a complicated combination product calls for sophisticated technologies, and so here's just an example of how we are incorporating AI into our technology to allow us to control all of these parameters and monitor them in real time. And so on the left-hand side, you could see a visualization of that fiber with the cells loaded in the fiber being created, and we're able to monitor the critical parameters. And on the far right-hand side, you could see where along the length of that tissue you're looking at, and at the end, you're essentially able to get a report. And so really, we've called this quality by machine vision, and as a sophisticated technology that we've had to develop to meet the complexity of what we're having to do. And this is, of course, a big part of discussions that we've been having with the FDA. And so a big initial application that we're focusing on is type 1 diabetes. Many here are aware of this debilitating disease, an autoimmune disease that relies on exogenous insulin injections to maintain blood glucose. It's a big challenge, and our goal here is to develop a functional cure by creating an islet replacement therapy without the need for immune suppression. And so that is our goal as far as it relates to type 1 diabetes. And so here what we can see is data from multiple different animal models. We've used the gold standard STZ model and demonstrated robust blood glucose control, both in immune deficient animals and immune competent animals, both in a xenogenetic context as well as an allogeneic context. So this gives us, of course, big signs of encouragement in terms of the preclinical data that we're able to generate and efficacy that we're able to showcase. And so a couple months ago, we were very excited to announce a major partnership with Novo Nordisk. We're very proud to work with a pioneer in the diabetes space. And so we're working closely with Novo Nordisk to develop up to 4 products in diabetes and obesity. And so, of course, Aspect is looking to leverage our proprietary bioprinting technology, and we're working side by side with Novo Nordisk, who brings incredible capabilities as it relates to stem cell differentiation and cell therapy development and manufacturing. And so, again, we're incredibly excited to have chosen them as a partner, and we look forward to keeping the industry updated as we continue to make progress on this exciting partnership. Another big area that we're focusing on as far as it relates to our proprietary pipeline is liver. So liver is an area that we're very, very excited about and essentially creating responsive tissues that perform metabolic functions inside of the body that correlate to specific liver disease indications that we're evaluating. And so from a high level perspective, there are two big areas that we're focusing on. The first is acquired liver diseases, specifically acute liver failure and acute on chronic liver failure, as well as congenital liver diseases, where we're looking at replacing specific functions that are lost due to these hereditary liver diseases. And so I'll highlight one of these programs that we're advancing forward towards clinical evaluation over the next couple of years, and that's in acute liver failure. And so what we have here is a demonstration of efficacy in an immune-competent animal model of acute liver failure. And so in this animal model, we've used the carbon tetrachloride induction of disease. And so for this particular disease, ALF, a patient is presented with a failing liver, and if they don't have a transplant within a couple weeks, they unfortunately in most cases won't make it. And so our goal would be to implant a tissue that only has to be in the body for a couple weeks, and that would bridge to recovery, or in the case of patients that have an underlying chronic liver condition as well as the acute condition, it would bridge them to a transplant, which would be a big deal in and of itself in terms of being able to have more time to get a transplant and potentially having a better match for a transplant. So both trying to bridge to recovery and also bridge to transplant for patients with acute on chronic liver failure. And so again, in this animal model, this carbon tetrachloride-induced liver failure model, we were able to show that we have about a 67% increase in survival when we look at the arm that included our bioprinted liver tissues compared to the sham control that didn't receive the living cellular therapy. And so again, data that we're really excited about and that we presented recently at the ASGCT meeting, and we're moving ahead with advancing the development of this program. Of course, I want to highlight our amazing team. It's very easy for me to stand up here and share the incredible progress we're making, but it's really them that are doing incredible work behind the scenes. Several people that have done amazing things in biotech that have joined the Aspect mission, and so we're really excited about the team we put together, and we're actively hiring right now, and so definitely interested in speaking if you're also aligned with what we're trying to do here in terms of bringing in this new category of regenerative medicine forward. So just to bring it all together in terms of what we're trying to do at Aspect, we see this as the next step in cell therapy, looking at harnessing the power of cell therapy, but combining that with technology like 3D printing and microfluidics and our machine learning-based approaches to create actual products that harness that power of biology. And so our platform allows us to do that. It's a broadly applicable, flexible platform that provides both the development capabilities and the manufacturing capabilities to make these products. We're applying it across multiple different programs, starting off with metabolic and endocrine indications, with T1D being the lead with our partners Novo Nordisk. And of course, moving forward in liver with our lead indication, acute liver failure. Of course, partnerships are important for any biotech with a broadly applicable platform, and we're very excited to have an excellent partnership in Novo Nordisk. And last but not least, we're moving forward towards vertically integrating all of the key capabilities to support clinical manufacturing over the next couple years, and so we are building all the necessary capabilities to allow us to go all the way is our goal at Aspect. So with that, I'd like to thank you for listening to the Aspect stories. Really great to see so many familiar faces, but also look forward to connecting with new folks over the coming days. And so I'm happy to take questions offline, but I recognize I'm out of time, and I appreciate you listening to the Aspect story. So thanks so much.